Oh, just before we begin, Emilio and I wanted to remind everyone, all of the trainees, that to check out the applications for the OTS mentorship program, as well as travel grants. Both of those things are super important for trainees to look at. But yeah, my name's Sarah. I am a fourth year PhD student in the lab of Anastasia Karova at UMass Medical School in Worcester, Massachusetts, in the US. I primarily focus my research on Huntington's disease and pushing forward various oligonucleotide therapeutics for that. And that's what I'll be talking about today. But I've also been doing a decent amount of work in terms of trying to optimize sort of different oligonucleotides, including siRNAs and ASOs. Um, so the title of my talk is Designing Oligonucleotides to Selectively Lower HTT Isoforms. And like I said, most of my work revolves around Huntington's disease. Huntington's disease is an autosomal dominant inherited neurodegenerative condition. So it's caused by the inheritance of an expanded CAG repeat within exon 1 of the Huntingtin gene. And it's marked by most interestingly sort of this adult onset in most cases, which features a loss of motor control, cognitive and emotional decline, and sort of at the cellular level is marked by a loss of the medium spiny neurons in the striatum. And within those neurons, we see sort of large amounts of aggregated mutant Huntingtin protein. And that's kind of thought to be the key pathological feature of disease for a long time until we've sort of gotten more and more research showing that the disease is caused by this expansion in CAG repeats that expand over time. And it seems like those DNA repeats actually may be sort of an important causative factor, and that an increased number of CAG repeats and not CAA repeats, or which also encode for glutamine, so the same sort of amino acid sequence, are correlated with an earlier age of onset. So more CAGs leads to an earlier disease onset. And if we start to look at things at a molecular level, you see that a patient with Huntington's disease will inherit one wild type copy of Huntingtin, as well as their mutant sort of expanded copy of Huntingtin. And that copy is sort of unstable. And these CAG repeats go on to expand throughout the lifetime in this process called somatic expansion, and you get an increased number of repeats, more and more. And this happens in kind of a mosaic manner across the cells in the brain. So different neurons have different sort of loads of CAG repeats. And this triggers then not only transcription of that sort of mutant expanded Huntingtin, but also these alternative splicing events in which exon 1 and intron 1 are not spliced out and then polyadenylated, creating this other sort of transcript that's only present in the context of this expansion called HTT1A. And it includes the CAG expansion, exon 1, and a bit of intron 1 as well. Then both of these different RNA species are found within clusters of the nucleus of the cell. And both of these species are translated into protein as well. And it's thought that some combination of these various factors results in the neuronal degeneration and toxicity that we see in Huntington's disease. So, one of the main questions sort of in the field is, where is the threshold for CAG toxicity? So you can imagine you have sort of all of these CAGs, and we know that before 40 is not toxic. That's what's found in sort of a healthy individual. But at what point does it sort of accumulate toxicity? And various studies have looked at whether or not knocking down MSH3, a factor known to modulate somatic expansion, can sort of ameliorate disease phenotypes. So looking at the transcriptional profiles of an MSH3 knockout mouse with around 180 CAG repeats, you see no reversal of sort of transcriptional dysregulation or any impact on this mouse model from this knockout. But if we look at 140 repeats, where you silence the same modulator of somatic expansion, you see this really huge impact on the transcriptional profiles of the cell. And other studies looking at single cell RNA sequencing, tying back those CAG lengths to individual cells, showed that cells with over 150 CAG repeats start to have significant transcriptional dysregulation. So we've sort of come to this model in which there is a period of non-toxic expansion from somewhere around the inherited 40 CAG repeats to around 150 CAG repeats, and then there becomes some sort of point of no return later on, in which the cells have completely been dysregulated and are experiencing toxicity from the burden of these CAG repeats. So we kind of want to investigate what changes are leading to this transcriptional regulation. And we know that wild type Huntingtin in mice seems to be evenly distributed between the nucleus and cytoplasm of cells, as shown here in red. However, when you look at human Huntingtin, which in this particular transgenic mouse model contains that expanded CAG locus, we see that it is much more nuclear localized, as well as forming these sort of larger, more insoluble clusters. Seen here. So the nuclear fraction of human mutant Huntingtin is much higher than mouse sort of wild type Huntingtin in this mouse model. And you can see that sort of quantified across images here. You see both more nuclear foci and larger foci, indicating the formation of these mRNA clusters. So there's also an aggregation of mutant protein in the nucleus as well. This is sort of the more traditional thinking of the disease, that this mutant protein is accumulating toxicity in the nucleus. But we want to sort of try to investigate why this toxicity is occurring. And to do that, we use various siRNA to knock down different candidate genes involved in disease. So I'm sure this audience is familiar with siRNA, which functions through the RISC complex. It associates with AGO2 to then sort of cleave and lower target gene expression. In the Corova lab, we use this divalent siRNA scaffold for CNS delivery. This is two siRNA linked by a tag. They're asymmetric and fully chemically modified. So we use 5' vinyl phosphonate modifications, as well as 2' fluoro and O-methyl ribose modifications, phosphorothioate backbone modifications on the ends, and this 3' exon A modification, which was developed in the lab. And these all work to sort of prevent some of the problems associated with siRNA, such as immune activation, but lack of stability, and make it a better drug. So we see that this divalent siRNA works really well in the brain over time. So our divalent siRNA against full-length Huntingtin silence is accumulating in the brain at up to 6 months post-ICV injection. And then we also see that it's lowering the mRNA significantly while it's accumulated, again, up to 6 months. And it's lowering the protein even more so at 1, 4, and 6 months in some regions. So we kind of applied this full-length Huntingtin lowering to different mouse models and looked at the differences in silencing between mutant and wild type Huntingtin. And this is cross-reactive, so it should work against both mouse and human Huntingtin. And in this transgenic model of Huntington's disease, which contains one copy of mutant human Huntingtin with around 120 uninterrupted CAG repeats, and then the endogenous mouse Huntingtin, we treated with the full-length Huntingtin targeting siRNA or an NTC, and then we measured both mRNA and protein. So looking at the silencing of mRNA with this siRNA, we see around 50% lowering in both the striatum and the cortex when we look at specifically the wild type Huntingtin gene. So this is the mouse copy. When we look at the human copy, we see only around 25% silencing, and it is in fact not reaching significance. So we kind of began to wonder why this was. And we then looked at sort of the localization of the mRNA. And if we look at this with fish, you can see the cell nuclei here in blue with DAPI. And then in red, we have the wild type mouse Huntingtin, so that unexpanded copy of Huntington. And in green, we have this expanded human Huntington, and you can make note of the clusters in the nuclei once again. So we saw that when we treated this with our siRNA, we saw nearly a really strong reduction of the mouse Huntington mRNA. And the remaining sort of foci seem to be predominantly in the nucleus. Then we look at the human expanded Huntington, and you see much less silencing. And you also see that these nuclear clusters are largely still present. So when we just look at the quantifications of this, we see wild type mouse Huntington is really strongly silenced in total. In the cytoplasm, it's even better. And then in the nucleus, we still see significant silencing. If you look at the human Huntington, we see less total silencing, but still significant total silencing. We see strong silencing in the cytoplasm, and then no significant silencing in the nucleus. So our conclusions are that this siRNA against full-length Huntington is not able to disrupt the mRNA that is clustering within the nucleus. So what we wanted to analyze is whether there's another transcript targetable by siRNA that might be seeding this Huntingtin aggregation. And just returning again to the molecular mechanisms, what other transcript do we know is present in these clusters? We know that this HTT1A exon 1, intron 1 fragment is heavily present in these nuclear mRNA clusters. So here we look at HTT1A lowering, and this is data from one of our collaborators in the UK, and they see in their mouse model that they lower HTT1A just barely in the hippocampus. This is not significant. But when you look at their protein data, you see significant lowering of the HTT1A. And then when you look then at the protein aggregation, we see nearly wiped out aggregation within this region. So a non-significant silencing of HTT1A mRNA leads to a significant silencing of HTT1A protein, and then downstream completely reduces the aggregation within this region. And what we wanted to know is whether a more robust knockdown of HTT1A was able to disrupt the presence of these HTT1A mRNA aggregates. And to do this, we've screened a lot of siRNA. So this is just kind of a map showing every sequence that we've looked at. And we've screened other classes of molecules as well. But in red here are just all the siRNA we've screened and all the positions in the intron. And we, looking at the silencing data, we get a number of different sites that seem to effectively silence HTT1A in mouse models. And then testing the dose response, we see that these are sort of dose dependent as well. So we get really nice dose response curves using our siRNA against the HTT1A. So this is our first sort of identified hit. And here we see that it works somewhat well, but this is sort of the first one we looked at. So it was kind of our target to beat. And then here we, these are sort of the next three that we selected to go in vivo. And these are, so these four are sort of our candidate siRNA that we chose to inject into a mouse model of Huntington's disease. So here is the in vivo data. So this was through ICV injection. In the gold, it was sort of our top candidate previously that had worked in vivo. And then these are our new candidates in red and green. And we see that the new siRNA performs significantly better than our old siRNA against HTT1A within the striatum, which is the most relevant brain region for Huntington's disease. And then in blue, we have our sort of positive control siRNA against full-length Huntington, and kind of, I think somewhat interestingly, we kind of recapitulate the same things we've seen before in terms of kind of a medium silencing of human HTT and a really solid silencing of mouse HTT. So this is definitely reassuring to see. But yeah, so this is all kind of ongoing research, and we aim to sort of start these long-term studies looking at HTT1A lowering over time in Huntington's disease mouse models, in which we can look at various biological readouts of HD. So we'd like to look more at the RNA clustering. This is something we've partnered with another lab on. They have an expertise in sort of imaging and microscopy and analysis, and they're helping to sort of improve the fish data you previously saw to be able to get sort of a photon-based measurement of these RNA clusters and get a better understanding of the RNA content of them, whether or not we're sort of changing them at all, and whether or not there's any sort of difference in cluster size or disruption occurring with siRNA treatment. We also aim to look at sort of protein aggregation. We've already seen this strong disruption in protein aggregation from HTT1A silencing. So we would like to look more closely at whether or not with a better silencing, we can see a decrease in protein aggregation, especially while looking at the human transcript. The previous data was just looking at the mouse version of this transcript, but we'd like to look at whether or not we're able to silence human HTT1A and get rid of human protein aggregates, as this will better sort of inform disease treatment. We'd also like to look at sort of histopathology, just looking at sort of immune activation within the brain and whether or not we can get, we alter sort of any HD phenotypes by playing with the expression of this HTT1A molecule. And then lastly, just sort of as a readout that a lot of people are interested in seeing, but can be a little unreliable, we'd like to look at mouse behavior. And I think it would just be really interesting to inform whether or not we're seeing any transcriptomic and behavioral changes when we knock down this transcript long term. But these are all sort of next steps in setting up long-term studies with these molecules that we've kind of shown work fairly well already. And with that, I would like to thank the rest of the Korova lab, as well as our collaborators in the Aronin lab at UMass, as well as the DeFiglia lab at MGH, and the Bates lab at UCL and at CHDI. So yeah, I guess with that, questions, or we can transfer to Emilio.
Yes, thanks a lot, Sarah, for this excellent presentation and great work. So I will start with a question. If you have a question for Sarah, please use the Q&A or the chat function. So obviously, when you, I think that Huntington's disease is caused by a gain of function mechanism, I suppose. So the strategy that you are proposing is a knockdown of the Huntington protein. And I think always what's important. So what is actually the function of the Huntington protein? And do you care about the amount of knockdown? So what kind of percentage of knockdown do you prefer in the end? Do you want a 100% knockdown so that there is no mutant protein at all? Or are you okay with some leftover levels?
Yeah, so I guess a couple things to that. One is we really don't understand the function of the Huntington protein. It does seem like it has a lot of really sort of important roles in synaptic function and health. So it is kind of important that we try to find ways around fully lowering that wild type protein. So with knocking down sort of the full length Huntington, there is some thought that maybe a 50% knockdown would be better. Maybe fully knocking down that protein is not ideal. The other sort of thing is that there are different targets. So if we, for our HTT1A project, we specifically are targeting the intron. So with that, we should see no lowering of the full-length Huntington by siRNA. So that kind of creates a more disease-specific way to interfere, as well as there's other projects in the lab looking at modulating somatic expansion. So they play with mismatch repair proteins in order to sort of prevent that CAG from getting bigger over time. And those don't interfere with the expression of Huntingtin at all. They're just interfering with the expansion process. So I think you're absolutely right. There's a lot of considerations to be made for the fact that that wild type protein seems to be doing something that could be important. But I think that's why we've been trying to come at this from all angles and develop a lot of different siRNA that sort of can approach the disease in different ways.
Yeah, yeah. So I think maybe, and I think also the Karova lab might be working on that as well. Is what about allele-specific silencing? Because I think
Yes.
also when you look at I think it's Wave Therapeutics, they are also working on that. So do you see a future for that kind of strategies?
Yeah, I think allele-specific silencing is certainly the way to go if you are aiming to develop a full-length Huntington silencing mechanism. I do think a lot of our work and other work in the field is starting to show that maybe Huntington silencing isn't the way to go at all, in that some of the work that I've seen from collaborators and our lab shows that these mismatch repair modifiers seem to be even better at reducing sort of the transcriptomic phenotypes than Huntington lowering alone. But yeah, I think the allele selective stuff is definitely better than just knocking it all down. We do, I believe, have an allele selective compound in collaboration with a few other labs. But yeah, there is still kind of an outstanding question in the field of whether targeting Huntington is the best way to do things, whether Huntington lowering is the best strategy.
Yeah, so to add to this, I think the focus of the field, I guess, is moving towards the somatic repeat expansions, and I think you showed kind of the point of no return when you have 130 or 150 or 180 repeats.
Mhm.
So how can we use this as a biomarker? Because I think at a certain stage you need to have like there are lots of natural history studies done, and we know a lot about the disease progression and the history of these patients. But it might be actually quite challenging to determine the somatic repeat expansion size within neurons or specific neurons in the brain, right? So any thoughts about that?
Yeah. Yeah, no, it certainly is a challenge. I think there's a lot of work right now on on ways to try to measure somatic instability in the blood or CSF or other peripheral markers. I know there's other groups that are very focused on those biomarkers because it's so important for developing any sort of clinical trials and meaningful outcomes. And that's also, yeah, as you said, a big challenge for the mismatch repair sort of expansion modulators. Being able to prove target engagement is a major question, and so there's not a strong way to be able to do that yet in a biomarker. But I know a lot of labs are really working on getting that somatic expansion measurements in the periphery or the CSF. Yeah.
So and the other thing is, so seems to me that the intron one is a toxic species. So to say, I think
Mhm.
this is kind of what you presented, but you still have the repeat structures on the DNA, but also on the RNA level. And if I remember correctly, that also these on an RNA level, the RNA folds in specific structures and also forms RNA aggregates or um, RNA foci, which also can attract a lot of RNA binding proteins and other things. So also that might be part of the cause of the disease. And the second thing is that also it's not only the that, but I think that also and that has been shown, especially for the juvenile cases, is that the repeat itself is translated into other proteins. So do you think that your strategy also might help for that, or do you think that we have to worry about it at all?
I do think it's a worry and one that's been largely, I think, underexplored. We, yeah, we're really trying to see if we can modulate that RNA structure in any way, and it's certainly been something that's very difficult to do, which is, I think, why we're trying to just get many approaches to see if any of them are able to modulate that structure, and that CAG as well. I think there's been a lot of work by different groups trying to develop ASOs that specifically target that CAG tract, and with varying levels of success. It's a very difficult thing to do. It's such a structured region, and it's very long and it's very CAG heavy, and there's a lot of different CAG tracks throughout your genome, and there are different CAG repeat disorders that can occur. So trying to maintain specificity while targeting those CAGs is very difficult as well. But it is something that I think we're trying to work on, as well as other groups are trying to work on, is seeing if that's a way, if CAG targeting would be sort of a way to interfere with disease. But I guess we're focused on trying to see if we can get the clusters, and then sort of whether or not we get the clusters, a lot of people do seem to think of it as a very protein heavy disease. And if we can get rid of the protein and we can observe outcomes, do we then see strong modulation of HD phenotypes with protein silencing alone, or do you need those RNA cluster silence as well? So I think we're hoping to uncover some of the biology with this, as well as continue to develop new technology that we can use to look at different aspects of disease.
Yeah, I think that's the right way to go. One last question, maybe. So I think you already mentioned that, especially in the Corova lab, you are developing a lot of different strategies. So not only targeting the mutant protein, but also the mismatch repair pathway. So can you envision combination therapy, where you do not only address or attack the somatic expansions, but also the protein itself?
Yeah, so I think, yeah, that paper's published. There, we already have a paper on sort of a bivalent siRNA, so that divalent siRNA, but instead of two of the same RNA, it's two different siRNAs, so it has sort of two different targets in one molecule. And there's a lot of efforts in the lab to expand that. So can we get targeting of 3+ compounds? Can we link different modalities together? How much can we combine? There's certainly been a lot of effort that we've done with combinatorial therapies, as well as trying to develop new molecules that would be like a unimolecular compound to get multiple targets in one. So yeah, that's stuff that I and other people in the lab are actively working on, and hopefully I'll be able to share some of that data in coming years.
I was curious about one more thing. So
Oh
what kind
yeah.
of behavior test are you going to do? So it can be a short answer.
So there's another lab here at UMass that the PI worked under another PI who developed this really interesting technique called motion sequencing. It's kind of an open field test where you put a mouse in a box and you record your mouse in a box, and it then used sort of an AI approach in which it then analyzes the mouse's behavior and separates it into different syllables. So it's kind of an unbiased way of analyzing how that particular mouse is behaving and moving in just in its little bucket. And then it sort of analyzes that syllable usage and different behaviors to see if the mouse is behaving differently. And it seems like a very robust way to sort of unbiased analyze like a large series of mouse behaviors. So we're hoping to employ that approach, as well as just sort of the more typical nesting, foot grasping, hopefully not too much rotor rod, but just typical behaviors.
Oh, cool. Sounds like a super interesting approach. So I give the word to Aaron.
Great. Well, thank you. That was an awesome talk. And really exciting to see what you're doing and all the kind of different stories that are coming out of these collaborations. And I really appreciated too that you suggested that, you know, maybe there's a different path than the path that we've been focused on for Huntington's disease for such a long time. That's really cool. So thank you, Sarah. We're going to move on now to Emilio and let you, Emilio, get yourself set up.
Yes. Hello, everyone. I'm Emilio Harris-Mostert. I'm a PhD student in the lab of Ipe Elgersma in the Erasmus Medical Center in Rotterdam at the Clinical Genetics Department. And I'm working on basically developing ASO therapies for rare diseases, for genetic diseases, which in the end we would like to apply in an NS1 setting. In this talk, I want to focus on kind of one of the projects I'm working on. And basically what this title is trying to say is that I want to make the computer choose the sequences we're going to be targeting with our ASOs. And why would you want to do that? Because if you have a gene and you would want to knock it down with an ASO, there is quite a lot of possible ASOs that could do it, quite often in the hundreds of thousands of ASOs that could be the one that knocks down this gene. But you can impossibly never screen all of these. So what you want to do is kind of narrow down which target sequences are actual potential targets for us. As a starting point, luckily there is a really nice paper published by Lieke Pedersen that kind of sets out this R script that does a bunch of stuff to kind of help choose which target sequence. And I really like that work, but I thought there's quite some other stuff that I would like to add to it. So let's start with what it already does and what my ideas are about that and how I'm kind of changing that, but also what I'm adding on to it and how I kind of envision this tool in the future. So let's start with RNA accessibility, which is a really important part, because to get your ASO in blue, or darker blue, bound to your target, it's not just, it's complementary, so it's going to happen. In the cell, there's a bunch of stuff happening. So to have both of them unstructured and close to each other, you need not only your target RNA to be unstructured, but you also need your ASO itself to not be binding to kind of itself or to other molecules of itself. So what the script does already is that it has a thermodynamics-based kind of prediction of these accessibilities and kind of the binding with itself. However, there might be some ways to actually start improving this. And one of them we're exploring right now is to use chemical probing data sets, because there is quite some chemical probing data sets of just really probing in cells, like how structured are certain areas of RNA, and can we kind of incorporate that data to have a better prediction of not only the thermodynamics, but also what the effect is of all the RNA binding proteins and all the other kind of factors that influence the structure of RNA. And other things we would like to kind of explore a bit is if we can improve this prediction using either machine learning tools for RNA structure or different types of modeling, like coarse-grained modeling instead of thermodynamics-based modeling. But that's all kind of still work in process. Right now, we still are using this thermodynamics-based modeling. And something else it also already does is it kind of takes a quick look at how many off-targets are there in the cell. The big problem is that off-targets are really difficult to quantify for ASOs, because not every sequence that is 100% homologous to your ASO is actually going to be an off-target. But also, sometimes there's going to be 1, 2, or even 3 mismatches or bulges within your ASO to the target RNA, but then it could still be an off-target. So just counting how many kind of predicted sites there are is actually not enough. But that is kind of the level we're at right now. It just looks at the entire genome and it sees for every sequence that could potentially be a good target for our target gene, how many off-targets are there within the entire genome. So already working at kind of narrowing that down for just RNA expression and just ideally having it tissue-specific, because in our lab, we're mostly working on the brain. So for the brain, it's quite nice because it's kind of a closed-off organ. However, if in the end you're going to be using ligands or other ways to kind of directly get your ASO to a specific tissue, it might be also nice to be able to change that for other tissues of interest. And something else we're exploring is if we can actually weigh the predicted off targets, because not every off target is actually a problem. You can knock down some genes and it won't actually do anything, especially in certain organs. So what we're looking at is either maybe looking at expression levels of genes, so not only looking at tissue-specific RNA, so which RNAs are expressed in the brain, but also like how much are they expressed, because the lower expressed ones might not always be as important. Or look at something like PLI score, which is a score that kind of predicts how kind of sensitive a gene is to being knocked down. Also, we'd be interested in doing an OMIM search of predicted off targets, because then you can already get an idea if knockdown of a certain off target is actually a problem and could actually be disease causing, because you don't want to kind of treat one disease and cause another one in the meantime. And finally, something that could help us predict which of the predicted off targets are actually going to be off targets is to also look at the accessibilities of those sequences that are homologous or have one or two mismatches with our ASO, because that could help us kind of see whether or not this off target is actually going to be knocked down. So now let's take a look at some of the things I'm considering to add to this script. So what are the sequence features? Because every sequence is, of course, its own, and just some features of the sequence seem to really affect ASO tolerability and efficacy. So first one, because we're looking at the brain, for us, we thought it really important to actually look at if this ASO is tolerated within the brain. And for that, we're looking at the acute neurotolerability score, which was published by Peter Hagedorn from Roche, because what they found is that if you inject ASOs into the brain of mice, some of them just in hours after that injection will suffer a lot from just getting injected by an ASO. So for us, because we work in the central nervous system, it's really important to also incorporate that score. Something else is that we're looking at the RNase H cleavage preference, because although RNase H is known as a protein that will cleave any DNA RNA heteroduplex, because that's the mechanism of action of our ASOs, having RNase H cleave the RNA targets, there seems to be some preference for cleaving certain sequences over others. So if you can choose targets that have kind of a sequence that is preferable for RNase H, then we increase the chance that if our ASO binds to the RNA, that there's actually going to be cleavage happening by RNase H, which is, of course, preferable if you want to have an efficient knockdown. And finally, there are some sequence motifs that have been published that kind of correlate to ASOs that are kind of better working or worse working. And one of the reasons we kind of look at this is because, for example, this GGGG unfavorable motif has been known to be very structured and be difficult to get into cells. And also, once it's there, it kind of doesn't bind that well. So that's one example of something that we would want to avoid. But for the other kind of favorable and unfavorable motifs, it's not really clear why they would be more or less favorable. So we prefer to have something like this cleavage preference or the tolerability score to kind of guide us. However, if these motifs do seem to kind of really help us choose target sequences that are better, we are going to incorporate it as well. But for now, it's just something that we look at the side, kind of do the sequences that we're actually testing have these motifs, yes or no, and then we're going to be kind of looking back in the end if that did have an effect for our kind of predictions, yes or no. So now we have a script kind of that's already looking at quite a few things. However, kind of designing ASOs doesn't end at that. And I just kind of want to give an example of how important it is to also look at other things. And one of those is the binding affinity, because just changing the binding affinity of your ASO is also going to really increase or decrease how good your ASO will work. So as an example, just kind of to test out how this script was working, we designed a bunch of ASOs, or actually 3 at the start, to target firefly luciferase, which we use as a kind of a readout protein for one of our screens. And we saw that when we test these ASOs in our screening method, that not all of these worked quite as well. So this second ASO actually seemed to not really cause that much knockdown. So we wanted to see, like, can we find out why this is? Because based on all the kind of factors from the script, it should be a good ASO. So one of the things we wanted to check is it the binding affinity? Unfortunately, there's not a good method to predict binding affinity kind of publicly available online. So what we use is this website that kind of predicts binding affinity based on LNA2O-methyl ASOs binding to RNA, because we feel just DNA, RNA doesn't really capture the effect that these chemical modifications have. And because we do already have LNA, we thought if we have LNA, DNA, but then LNA2O-methyl, it might get a bit closer to what the actual is. However, because we don't have the DNA, RNA binding that is a lot less strong binding, we know for sure that we're greatly kind of overestimating how strong these ASOs are actually binding to the target. And if we plot these 3 points, we see something happening, which is something that has been described in literature as an inverted, actually as a U-shaped curve, where it's been found that if your ASO binds too strongly, it gets less efficient, but also if it doesn't bind strong enough, it's also not that good. So you really want to be within this kind of range. So you were wondering, like, is this actually the reason that this ASO is not working well, or is there a different reason? So what we decided to do is to kind of decrease or increase the amount of LNA nucleotides in the wings of our ASOs. So what I did is I just kind of had the same graph as just now. I made it a bit wider, and now we're going to be adding a data point to the left and to the right of each 3 of these ASOs. However, what we see is that this blue ASO, this ASO2, it just doesn't seem to work. It just also shows that the script for sure is not going to be 100% correct in choosing target sequences. However, we do see kind of the start of this U-shaped curve being created for ASO 1 and 3. So I guess we can even continue adding more LNAs to see where actually that optimum binding affinity is. However, it does show that if we would have kind of reduced the LNA modifications more, which I guess for both of these would not have been a candidate ASO because you want more than one LNA wing, in the wing, we might have said, oh, this ASO doesn't work that well, even though it does work better if you just change the binding affinity. And finally, this script is actually really still a work in process. So I would also really kind of be happy if people watching now or later would have input either through an email or whatever. So if you scan the QR code, which will also be up in my acknowledgement slide, I would be really happy to discuss more what we're working on here and what your ideas are of how to improve this. So before I go to the acknowledgements, I want to highlight quickly basically the same things that Sarah also highlighted quickly, is to please apply for the mentorship program. Because it is a really good program, and it's really nice to just have a mentor within the OTS that is knowledgeable and that can really help you in your work, but also in your career. I think applications close April 30th for this kind of mentorship session. And also, if you're interested in going to the OTS in Budapest, please apply for a travel grant if you have some nice work to present, because I think it's really nice to have as many trainees present as we can possibly get. And also, of course, I think Erin and Ronald are doing a really good job with these webinars. And currently the trainee spotlight series is still going on, and I think the next one will be April 10th. So if you're interested in all the cool work the OTS trainees are doing, please join these as well. So with that, I would very much like to thank my lab, who kind of really helps me a lot in all these projects, and a special shout out to Jeremy, who was a bioinformatics student that kind of helped working on this project in the last few months. So thanks a lot for listening, and I'm happy to answer any questions.
Cool. Well, thanks a lot, Emilio. Excellent presentation. So before we start with the questions, I'd like to emphasize that because I've been in the OTS mentorship program, both as a mentee and a mentor, and I must say I really I enjoyed it. So it's also nice to talk to others and see how others experience their not only their career but also future perspectives. You can discuss things like how to write a paper, how to move forward, how to discuss particular things with your mentor or with your mentee. So I think it's really worth in joining the program. So and I think one more thing, I think if you join the mentorship program, you get a very nice OTS coffee mug. So that's also another reason to join the program. Or not. I'm making this up. I got one last year.
I think you get a free lunch at the conference too, right?
Oh, and you get a free lunch. It's even better. Yeah. It's super nice because you get a free lunch with all the big names in the field. But okay, let's focus on the work a bit. So a very nice presentation, and my first question, and actually I don't know myself how to do, because you discussed the off-target effect, and I think what you mean actually is the kind of on-target, off-target effect. Because
Yes.
when you treat, because you also have like secondary effects because you modify your protein of interest, so the downregulation or splice modulation. So how exactly do you determine your off-target effects? What kind of method do you use?
Yeah, so no, I completely agree. I should have been more specific. It's sequence dependent, or actually hybridization dependent off-target effects. So it's really just the nucleotides binding to other RNA just because there's enough complementarity. So what we do right now is actually what was kind of already in the script. It's just looking at the entire genome and seeing how many times does this, if I go to, so if we are, it should go all the way to the back. If we look at every single one of these options here, how often does, for example, this sequence or that sequence just come back in the entire genome? And how often do you find that exact sequence with just one mismatch? And it just kind of spits out the number. And then if the number is too high, we filter it out. So I think in the paper, they advised for the completely complementarity to just allow only one, which will then be your on target. And for one mismatch, they cut it off at 50. It's just kind of a number because you have to choose a number and it's not really based on much yet. But that's why also we want to really kind of improve this method, because it's really hard to kind of know, like not every off target, or at least kind of hybridization dependent off target is going to be just as bad. So that's why we really want to kind of include also looking at like what are the off targets, and can we kind of say like, okay, this one has quite some off targets, but none of these off targets really seems to be kind of like doesn't raise any red flags, so maybe we can still allow this ASO. However, if another one only has one off target, but that off target is a super important gene, it might not be a good idea to continue with that ASO. So it's really tough. And I think actually this paper by Anderson, which was released this year by it's kind of industry recommendations for dealing with hybridization dependent off targets. And it gives a really good overview of kind of what considerations you should make, how you can kind of build a script around different kind of methods that are out there. So if this is something you're thinking about and struggling with, I would highly recommend reading actually this paper, because it really gives a really good insight and overview on things you should be thinking about.
Thanks. Yeah, so you're mostly discussing, of course, the in silico approach.
Yes.
Just browsing through the genome. And I really like the idea of giving a little bit of more value on the super important genes and give a little bit less value on the regions in the genome that we don't know that much about. Because it might be very helpful. And another strategy, and I think it's also highlighted in the, or maybe not, in the yellow and the green part, is that sometimes what might be helpful is when you have like a one base pair mismatch with a very important gene, you actually want to introduce a second mismatch so that there is way less affinity with this very important target. Because I also assume, especially for splice modulating, for example, you don't have much wiggle room to move your ASO to the left or to the right. So is this also something that you want to include in your script?
I think for the script now, kind of the focus is really for RNase H dependent and then just the entire genome, like just the entire gene knockdown. We're also kind of one of the things it also does that I didn't highlight now is it looks at whether or not there's going to be a common SNP within your sequence. And in general, we kind of filter that out because if you want a common, like a general knockdown, you don't want to only be knocking down 50%. However, sometimes if you actually want to be allele selective, then actually filtering in those SNPs is a great idea. And I think for then it's more in following steps that it's really nice that if you have a good ASO, but you have problematic off targets or even just the other allele being the problematic off target, adding a second mismatch is something that I think has been shown to work quite well. So yeah, for us, it's definitely something that we do if we need to do it. We haven't done it that often yet, but it's something we definitely consider. Yeah.
Yeah. So you discussed next to this, you discussed the I would say the sequence features. So any guesses on the favorable sequences? So I can assume that when you have like 4 Gs in a row or something like that, that might be very unfavorable. But what about the favorable motifs that you show here? Are there any like common denominators, or do you have any suggestions?
I've tried to, for example, look at what if I add these motifs in, for example, the RNase H cleavage preference, but it doesn't really seem to kind of immediately jump out that, yes, this motif easily fits within this cleavage preference of RNase H. So it's that's not one of them, I think. I think one thing that really seems to jump out is that quite a lot of them have, well, actually all of them have at least 2 Cs, and in the stronger ones, like the biggest hits, even have 3. So probably there's also has something to do with just binding more strongly to a target in a way that doesn't kind of cause bad binding within itself. But it's, yeah, for now, for me, it's really still, like also in the paper, it was really a black box. They also try to kind of think about it a bit, but it's, yeah, I'm not completely sure yet why these would be more favorable than others. I think it's a great question.
Okay. No, thanks. Question from Damon in the chat. So great talk, Emilio. So I agree on that. What algorithms will you recommend for ASO or siRNA design? So
yes, I think for siRNA, there's a lot more out there. I personally haven't checked a lot of them, but I think a lot of websites say, like, just based on microRNAs, these would be good targets or not. I think maybe Sarah probably has a better insight in which of these are actually good algorithms or not. For ASO, I think I had a slide in the end of the most that I could find and what I did or did not like about them. So I think this is PF read, but it's also kind of in a paper it says it's actually mostly based on siRNA. And for me, I have some problems with enumerating it, and also it only gives me a number and it doesn't really kind of show how you get to that number. So I personally prefer kind of the paper I presented here, because it really gives you insight in why is it filtering out certain sequences and why is it filtering in other sequences. Then you have this LNC-ASO. It's based on long non-coding RNA knockdown, and I heard it's really good. Unfortunately, the website is no longer working. It was at one point in the OTS recommended tools. However, I've heard that maybe in the N1C, there are people that still have access to it. So I think if that's something you would like to try, those are the people to kind of email within the NS1 collaborative. And then recently also there was this ASO optimizer, which I think did quite some stuff that was interesting. However, it kind of, because it made like a neural network prediction of the best chemical modification pattern and kind of what the best structure or target was, but it trained itself on data for just one target, and then it also gave better ASOs for that same target. So I don't think it's very generalizable for knockdown of other targets. And also kind of probably you can train a model yourself using the same method, but then you already need a lot of data on ASOs targeting a certain target at different locations. So I don't think that one also is kind of in at least academic setting where you just don't really have data yet and you want to get started on a new target, I don't think it's the best starting point. So for me, what I really found is that this paper that was published by Lieke Pedersen, for me was a great starting point, and up to now I've had some pretty nice results. For sure, like when we're at a point for our script where we feel we've added quite some things that we like, and we're getting still good results, we're for sure going to want to make it publicly available, but unfortunately we're not quite there yet. And also we really want to be sure that we're actually designing good ASOs with this script before bringing it out there, because otherwise we would only be hurting the field if we kind of publish a script that's actually not working that great. So like up to now it's been looking good, but like once we get to a point where we're comfortable with what we have, for sure we will publish this and make it available.
Yeah, and then about the toxicity part, so I think you showed this neurotolerability score based on the Hagedorn paper,
Yes.
but obviously that's only one way of toxicity, and it's a neurotolerability assay, so probably it's
Yes.
mostly for brain disorders or ASOs for brain disorders. So can you recommend any more general toxicity strategies?
Yeah, I think there's quite some literature out there, kind of, I think mostly for really the hybridization dependent. Like even this neurotoxicity, it's not completely clear yet what is actually causing it. There's quite some ideas, but like at least maybe people know, but I've not seen data that completely convinced me that this one reason was why this is a problem and what we should do. I think the main kind of takeaway from that paper is to avoid having Gs within the ASO. But I think besides that, it's really kind of managing a bunch of aspects. So one thing that people do is using less phosphatidylate because that kind of reduces protein binding, and then that sometimes causes problems, but also not always. So that's, I think, one thing that people do. If an ASO really kind of has too many off-target, like hybridization-dependent off-targets, sometimes just reducing the binding affinity can help, because then the effect of those mismatches could be bigger. But it's yeah, I think it's still quite difficult to really be sure once you have your ASO whether or not it's going to be toxic or not. So that's why it's really important that before in the end you go to clinic that there's like an extensive testing done. But I think it's more and more understood, and yeah, I think for now what our ideas are is to yeah, reduce phosphatidylate. I think is something that could help if you have a toxic ASO. And just, yeah, changing binding affinity, or like using MOE instead of LNA, or just reducing the amount of modifications in doing. And I think also sometimes if within your gap you change one of the nucleotides to like an RNA mimic, so an MOE, for example, then it also reduces toxicity sometimes. That's something else that's been published. But it's, for what I know, it's still a big question, and there's still a lot of different opinions. So yeah, I hope one day we will understand it a lot better.
Yeah, and that brings me to my last question, at least for the specific question. So, because also there are a lot of new different chemistries out now, right? So, how easy is it to insert or to combine these new chemistries within your method?
Yes, I think it is something I would love to add in the end. I think for now we want to really just ignore all of that and just have, okay, we want to get target sequences, and then in the end you yourself have to kind of decide how many chemical modifications, which chemical modifications, what's the binding affinity. So that's why also in like my graph I have this binding affinity in a dotted line, because I don't think this is going to be part of the script in the end. It's just something I'd like to highlight, that once you get kind of target sequences from this tool, you're not quite there yet. There's still a lot of things you have to change and test and find out. So I think in the end, for this tool, it won't really be in there yet. I think hopefully at one point there will be enough knowledge out there that you can actually get a script that not only chooses your target sequences, but also gives you chemical modifications, where you want them, how many you want, and all of that. That would be amazing. Yeah. Cool.
Thanks a lot. And then one last question for the audience. how to apply for a travel grant?
Yes, I think the applications are not yet open. I think if you monitor the OTS website at one point, I think also when the speakers are a bit more known and everything, like the invited speakers, and they'll start promoting actually like the conference more, there will be information on the OTS website. So that's the place to keep an eye out for. So I think it's the OTS website, and then there's a awards kind of thing in the menu, and one of those awards is kind of the travel grant, and then there it has a really expensive kind of overview of all you need to do to get to apply for it. Yeah.
Okay, well, cool. Thank you very much. I give the word back to Aaron.
Thank you.
Well, thank you so much, Emilio and Sarah. These were really great talks. It's really cool to see an introduction of your work. We're excited to have you on the board and see what you can do, especially to represent and advocate for trainees. And so our next, speaking of trainees, as was mentioned, our next trainee spotlight series is coming up April 10th. And then we have another one scheduled for May 15th. So if you're watching and you're interested in presenting, please reach out. And we have a few talks in between. So as always, you can check out the webinar website for OTS. And we look forward to seeing you at our next webinar.